Today we're talking about pulse oximetry. So we're going to go through a review of PPG technology, a little bit of pulse oximetry, their biological foundational function, as well as how we're going to be doing in the lab. So typically for the times that I have you in class, given that it's a holiday, I'm not going to see you, but we still have online. I like to run this reflection, which is a way for me to ask what things worked or didn't work in the lab. So I think as a reflection, I might want you to think of something that was very clear or that made sense when you were in lab, and also consider something that perhaps was not clear. And then I would also go through the quiz results. But since we're not in class, I'm going to skip that portion. Now, for today, the learning outcomes are going to be around having students being able to explain how the pulse oximeters calculate oxygen saturation, be able to describe the roles of the red and infrared light in determining the SpO2 levels, which we're going to be talking about, and then also identifying some potential limitations that might exist with pulse oximetry. So the plethysmography, and I always struggle with this word, but plethysmography is the measure of changing volumes within the body. It can be used for things as big as whole body volume approximations, as well as all organs. And it comes from the Greek plethysmo, which is increasing, and then graphene or graph to write. In the context of this class, we're going to be specifically looking at photoplethysmography, or PPG, which I will be calling PPG from here on out because it is a mouthful, which is a non-invasive optical method. It basically detects the volumetric blood changes in peripheral blood flow by alternating certain lights and measuring the response of those lights. You can use this, and again, we're going to explain this in a little bit more detail. We can use this to do some cardiovascular monitoring in wearable devices. I think everybody has an Apple Watch or a type of watch that has this very simple technology, I have to say, that allows for the monitoring. Something for this slide that I do want to highlight is the fact that you have two types of transmissions, or two types of light shining methods. You have the transmission mode, which light is going through the tissue, and you have a reflectance mode, which is light that is bouncing off the tissue. And we're going to go through all of these details. This is just a bit of an introductory slide. So why PPG? Why do we use this, particularly in the context of measuring blood volumes? So the most obvious application is just measuring cardiac cycle and heart rate. So things that you can extract from that are respiration. There are ways for you to approximate the respiration of an individual based on their heart cycles. You can also measure the blood oxygen saturation, which is what we're going to be doing in the lab. You can even, with that beats per minute that you can extract with PPG, you can test things like heart rate variability, if there's some type of clinical condition that is causing heart rate variability, as well as pulse transit time, which is that interval time, as well as general things on vascular health. If you're getting weaker readings, you can infer some issues with the peripheral blood supply. So the pulse oximeter specifically is one of those applications within PPG. You might hear it as SpO2 measurement or blood oxygen measurement. So this is the application that we're going to be focusing in this lab. So a little bit of background in the biology of how this works. It may or may not be, hopefully is not an introduction to the fact that hemoglobin is a protein in red blood cells that carries oxygen. And there are a lot of species of hemoglobin and you all will know the biochemistry way better than I would ever do. But the ones that are the main focus when it comes to pulse oximetry are two, are the oxygenated hemoglobin, HbO2, and then deoxygenated hemoglobin, Hb. And it is the properties of these two proteins that allows us to be able to approximate what that oxygen saturation. And hopefully, when I'm already talking about oxygenated, deoxygenated, hopefully already in your mind, gears are turning on how could I approximate the blood oxygenation of a particular individual if I have the amount of oxygenated hemoglobin versus the amount of deoxygenated hemoglobin. So for the biology, how does this work? Like, what is the biological or biochemical basis of pulse ox? And it's somewhat fairly simple. The oxygenated and oxygenated bloods absorb different portions of light, particularly in the near infrared region. HbO2, which is the oxygenated, absorbs more infrared light, while the deoxygenated hemoglobin absorbs more red light. And you can see here, for example, that the oxygenated in red has more of this higher wavelength, which is going to be on the infrared, whereas the deoxygenated is going to be higher on here, which is the red. Now, how do we make that calculation? How do we actually approximate that ratio or that percentage? So the way that we can do it is seeing here in this diagram, we see that we have this red signal, which is going to be, if I remember correctly, because I always mix these two, it's going to be the deoxygenated blood. So you have the AC, which is the alternating current or the alternating portion of the signal. So if you take the higher peak and the higher value, that's going to give you the AC of that signal. And the DC is the offset relative to 0, or wherever you're measuring, whatever your reference point is. And that itself is going to give you the ratio of the, I forgot it already, the deoxygenated one. And then you can do the same for the deoxygenated and calculate the ratio of those ratios. So that is what we have here. So we have the AC of red and the DC of red, and the AC of the infrared and the DC of the infrared. And in order to calculate SpO2, we have what is called the R ratio, or the ratio of ratios, which is the ratio, or the variability, if you want to think about it that way, of the red light versus the infrared light. And already, as I mentioned, this is going to give you a proportion. And we can go ahead and plug that into this equation to approximate what that SpO2 for that particular patient is. Now, if you think about it a little bit, where does this 100 and where does this 25 come from? And that is a very, very good question. The answer is experimental and actual, I don't want to call it trial by error or just testing, but it comes from experimental validations of a lot of this technology. So whenever a device like this is being calibrated, you get a bunch of people and you test them with a gold standard, which is an actual protocol that I don't remember if it will be in the reading, you might bump into in the reading that I assigned for the lab, in which they have a chamber that they deoxygenate the blood in a very controlled fashion, and they know exactly what percentage of deoxygenation is going on, and then they test a device versus that, and they're able to extract this equation. If you do some research online, this is the more standardized equation. It is not to mean that it is universal and that these numbers are not written in stone. But these are the ones that are most standard, and these ones are the ones that we're going to be used. But I think it's important to note that a lot of this experimentation and a lot of this device development relies on validation and testing from a medical device type of procedure. Now, what are the AC and what are the DC components of the signal that we're working on? We have the AC, which is alternating current. We're going to get into that a little bit more when we go into circuits, but you can think about this as the oscillating components of a signal. And the DC, you can consider them as the steady offset of a signal. So when you get into verifying what that particular DC offset might be coming from, you have a lot of capillary and vein offset of values, tissue, anything that gets in the way that doesn't change is going to offset where that measurement is going to be. The AC component of it is going to be what does change, which is going to be the arterial flow, basically the pulse, the in and out of the blood, or I mean, maybe not the in and out because it's not coming in and out, but the pulse of the signal is what's going to oscillate. So you'll notice that in this signal, there is a clear offset, meaning that the signal is from the ground goes up and from the ground goes up. That is all the capillary, vein, tissue, anything that can get in the way that doesn't change, while the AC component or the alternating current component is going to be anything that does change. So that's just something to consider whenever you're seeing this AC/DC type definitions. And when we get into circuits, we'll talk about the actual definition of AC and DC when it comes to circuits and electronics. But in this case, it's just the variability. This is a class exercise, but this is an example of how that equation can change depending on the device. One of the highlights of this exercise is particularly you thinking about what might be wrong with this particular statement. So think about this equation that was derived from a group of young volunteers, let's say college age students, because I don't want to say most science, but a lot of science is tested on college students because that's where the university does the testing. So think about who needs pulse oximeters versus in this particular case where this equation was tested, the top one, which population uses or requires or absolutely needs the pulse oximeter. And that hopefully should start making some connection between the testing, validation, and the need for robustness in how we calibrate a lot of these equations. And the answer is young people are going to be the least to use a pulse oximetry. You're going to have aging population. You're going to have very specific groups with diseases and disorders related to heart variability or even oxygen variability. So that already should tell you that there is a bit of a bias whenever it comes to testing a lot of these equipments and why there's a lot of regulations and standards, engineering standards that go into this particular device. One thing that I also like to introduce, particularly in this lab, in this pulse ox lab, is the fact that the circuit is not complicated. It might seem complicated, but it honestly is not complicated. In this diagram here, there's honestly a lot of things that are actually not necessary. In reality, you can get away with an Arduino and 2 LEDs and a resistor, something like this, for example. Not even these switches, which are, I'm blanking on the name for this component. It's not going to come, but you only really need a resistor to not overload your LEDs and an Arduino. So you can alternate the light and measure your signal. So it is an incredibly simple circuit. What these circuits are doing with these components, which I'll hopefully remember by the lab, or this op-amp that you see here, is that they're making the signal bigger. So it's a little bit more detectable. But honestly, it's not really necessary. You don't really need it. And in this case, this LM7805 it's something additional that again is unnecessary, and they use it for this little diagram screen. So in reality, you don't really need it just to get the results, mind you. If you want to make a device, yeah, you'll need it, but in this case, it's not necessary. Now, some clinical applications for the pulse oximeter. Given the fact that it's measuring heart rate, it can detect some heart attacks, heart failure. Specifically in the case of breathing or oxygen-related disorders, it can monitor COPD, chronic obstructive pulmonary disease, anemia, some lung cancers because of the diminishing returns of the lung due to the cancer, asthmas, or pneumonias. So anything that pretty much gets in the way of oxygen being in the blood. So limitations, this is something that I would need to do an exercise, but I recommend you to look up online, even if you want to do, you're in the computer because you're watching this, maybe you're on your phone, but just do a quick Google and look up one limitation of pulse oximetry. Like what can get in the way of pulse oximetry? And I'll give you like 5 seconds to just do that. And it's like those YouTube videos. I don't expect you to pause, so I'm not going to ask you to. One of those limitations comes in the effect of lack of calibration or lack of subjects for calibration that have darker skins. This is something that has been heavily investigated, and particularly during the pandemic, came into a pretty strong spotlight because a lot of individuals with darker skins were having inaccurate readings, and therefore we're not getting the care that was needed. So as an engineering problem, as a societal challenge, it's very important that we test pretty much everybody from one end of the spectrum to the other in order to make sure that we are designing, testing, validating, and calibrating our equipments with as much information as possible. And in the project, we're going to talk about a little bit of how we can do that through statistics and design of experiments. Some other limitations that I delete them from the last class can be weather, like temperature. If it's cold, the peripheral blood flow is not going to be as perfused. You can have nail polish. You can have pressure. Those are kind of the ones that come to mind. So this is a good exercise. I should have kept them from last class. Some potential solutions, some ideas that have come around the concept of, particularly for darker skin, is shifting the wavelength. Red doesn't seem to be as reliable as other wavelengths, such as green. You might notice that a lot of current devices actually use green light rather than red light. The awareness, bias, calibrations, different materials for the lighting. I believe some students found that you could use some combination of light and sound, or make sure that you're using very well-known areas of the body to get these readings from. So there are ways that, again, as engineers, you can fix and address rather important societal needs whenever you're designing a video. For references, I found this very good summary video if you want to give it a look. It just goes through the basics, a little bit more on the clinical perspective, but again, it's not a hard video to go over the basics of how this technology works. And as for the learning outcomes, we talked about the pulse oximetry calculating oxygen saturation, describing the role of red and infrared light determining SpO2 levels, and also identifying some limitations of pulse oximetry. So in the lab, we're going to be measuring the pulse oximetry using some boards that I purchased. So hopefully it's going to be fun, and we'll get to have some good time measuring information. So if you have any questions, just let me know.